Okay, so we're here today at the 86th Cold Spring Harbor Symposium on Biological Timekeeping. So my name is Luke Smith, and I'm an associate editor at PLOS Biology. And I'm very pleased today to be joined by Dr. Mimi Shirasu Hiza. So thank you for making the time today.
Of course.
Dr. Shirasu Hiza is an associate professor in the Department of Genetics and Development at Columbia University, where she's also co-director of the genetic approaches to development and disease graduate program. And her lab has been doing really interesting work on how circadian rhythms influence health and disease using Drosophila as a model organism. So Mimi, I'd like to start out by hearing just a bit about your background and kind of what initially drew you to study circadian rhythms and also sleep and how they influence physiology. So yeah, just a bit about your background.
I did my postdoc in David Schneider's lab at Stanford. Very creative guy, studies Drosophila immunity. And really I study circadian rhythms because I'm a circadian mutant. I think I have a long period. So I tend to, in graduate school, I would cycle all the way around the clock, free run, you know. In postdoc, I didn't have that luxury so much. I had kids when I was in postdoc, but I did work pretty late in the lab. And I went to my boss one day and I said, so I'm infecting these flies at different times of day and getting like completely different responses if I infect them at 2 in the morning versus 2 in the afternoon. And he looked at me and said, well, you're probably doing it wrong. I was like, no, I probably am doing it wrong, but there might be some biology there to study. He said, well, that's your biology because you, the experimentalists, are different at 2 in the morning versus 2 in the afternoon. So that's totally fair. I'm probably a better injector at 2 in the morning than at 2 in the afternoon.
Yeah,
and so,
I remember that.
Yeah, so he asked me, I asked him to help me, and he went to the hardware store and, you know, bought these boxes and hooked them up with lights and timers and helped me rig a couple of chambers that were in antiphase with each other in terms of light entrainment. And so I was able to do the experiment of injecting the flies at the same time of day for me, the experimentalist, different times of day for the flies. And sure enough, you know, same needle, same genotype, same wild type flies, everything, except the only difference between them was the time of day they thought it was. And we got a completely different survival phenotype. So the flies that were injected at night with Streptococcus pneumoniae survived a lot longer and often cleared the infection relative to flies that were injected in the afternoon. So that's, you know, that's wow. So that's when I realized that, you know, what a profound effect circadian regulation can have on basic physiology, including, you know, the immunity against infection. And I said, you know, I'd love to just work on this. And, you know, David is really great about letting people be creative and independent and, you know, follow up their instincts. So that's when I started the project and took it to my independent lab.
Yeah, that's
so now
a serendipitous thing, and I didn't realize that you had brought the circadian component to the lab, which is that's interesting. So can you tell us? So you mentioned that flies respond differently to bacterial challenge depending on the time of day. So can you tell us a bit more about like exactly, you know, what do you see in flies and how does their immunity change during the course of their circadian rhythm?
Right. So it actually turns out, at least in the fly, to be phagocytosis by innate immune cells or hemocytes in circulation in the blood, so to speak.
Yeah. So I was going to ask, what do you think the mechanism is? So do you think that the phagocytes are responding to some cue from like some top-down cue, or do you think
that is a really great question, Luke, and something that we chased for many years because we wanted to. I mean, the most basic question, of course, is like, you know, is it a clock inside of the hemocytes themselves, or is it the brain clock, or is it some other cue in their environment that is changing? And we still don't know the answer to that question. Other people have found that there is a clock in macrophages in mammals, and that clock is important for, you know, various various aspects of their function. Also, there's a lot of beautiful work done on hematopoiesis and circadian regulation of hematopoiesis. But for the flies, we didn't have a great tool, unfortunately, to knock out the clock in a tissue-specific manner until a couple of years ago. We developed a couple of CRISPR-based tools to knock out. So now we can actually do those experiments, and we're doing that now, just sort of knocking out the clock in different places and seeing what the effect is on different types of infection.
And do people see like a similar effect in other species? Like, is there a circadian rhythmicity to immune function in other organisms? I would imagine so, but I wonder if it's been shown or tested or.
Yeah, so for example, when humans get vaccinated at different times of day, the flu vaccine, then you can check their antibody levels, a couple of weeks later, and it's different depending on the time of day. People have done those kinds of studies. It so much about the human body is circadian regulated. I think it's fair to say that everything changes, one's physiology, but tying those changes in physiology to specific sort of effects on disease or defense against disease is actually way more difficult, just because the body is so complicated, and all of these physiologies sort of interlock. Yeah, but this gets to something that we were talking about earlier about aging, and whether or not it's actually the individual circadian rhythms and individual tissues that might be lost and contribute to aging, or if it's sort of a more holistic sort of dissonance between tissues. That's a really interesting question, and one that isn't known, but I think is a sort of a great idea and sort of hypothesis in the field.
Yeah, so in a related topic, so I know you're also interested in sleep research, and so I wanted to ask you a bit about your work on sleep and in defining kind of what is its importance and why do we need to sleep? So I know you've done some work in flies. I wonder if you could tell us a bit about that as well.
Yeah. No, I think the sleep work is really interesting because we don't know why we sleep. It's a kind of mystery. I used to tell my students it's the last fundamental mystery left in science, and until they started throwing it back at me. Like, well, we have to keep studying it. It's the last fundamental mystery in science. You keep telling us that. Like, sorry. Yeah, I had this fantastic graduate student, Vanessa Hill, who wanted to study sleep, and I thought sleep would be important for immunity, honestly, in flies. And so
my mom always tells me to get my sleep, otherwise I'll get sick.
Right?
Yeah.
If I don't sleep, then I get sick. And when I get sick, all I want to do is sleep. So it seems really intuitive. And it probably is connected in mammals, but what we found was in flies, we couldn't find a specific, a sort of a common phenotype in terms of defective immune defense. So our approach was super simple, very, very naive. We collected a whole bunch of diverse sleep mutants and said, this is a behavior that's conserved, right? Evolutionarily conserved for a reason. There must be some fundamental reason why, animals keep sleeping and wasting their day or their night without any contribution or obvious contribution to fitness, right? So how is this trait conserved through evolutionary time? It must serve a purpose. And so if we collect flies that are short sleeping, they'll be defective in that purpose, and they'll show a common phenotype when given similar stresses, right? So even though these were flies that were defective in, ubiquitination or specific channel proteins or, dopamine transporter. So they're very diverse in terms of the sort of molecular mechanisms that cause lack of sleep. They had one thing in common, which is that they didn't sleep very well. And so we thought if we inject them with all kinds of bacteria and look at, each bacteria being defended against by a specific set of immune mechanisms, we would be able to profile them and figure out which immune mechanisms were defective in that was common to all of these flies. And there wasn't one. It was like really messy. So I told my student that she should.
It must have been tough for the student, I imagine.
Yeah. I was like, you should move on, I think, because this is like not a hypothesis. I mean, I figured we were being very naive, which we were. But she continued working on the project. Last fundamental mystery of science left, etc. And she found eventually that all of the flies were sensitive to oxidative stress. So sources of oxidative stress that were very varied, like paraquat, which generates superoxides from mitochondria, it's a mitochondrial poison, or hydrogen peroxide, which is itself, reactive, or later we found hyperoxia would do the same thing. The short sleeping flies were always sensitive to oxidative stress relative to their genetic controls. So we then showed that if you increase sleep, you could actually increase defense against oxidative stress. And now we're just trying to find the mechanism or underlying mechanisms for that.
Yeah, I think that's really interesting. So a question I had when I was looking at your paper is so like from my limited, which is limited, I'll do a disclaimer, understanding of sleep research is oftentimes like in humans and things in mammals, it's thought that it helps for clearance of toxic or from metabolites from the brain. It helps homeostasis in the brain. And I wonder, do you think that this oxidative stress and like importance for sleep in dealing with oxidative stress is specific to certain cell types or tissues, or do you think that it's kind of an organism-wide phenotype where it clears oxidative stress in the brain and in the liver, and I mean, not that flies have livers, but like
no,
throughout the organ.
they do have livers. They have something called the fat body, which is sort of a combination of adipose tissue in the liver. Yeah, that's a great question. I mean, we would love to know where the buildup of ROS is. There was a paper a couple of years ago by Dragana Raguglia that was looking at this exact question and found oxidative stress in the gut. The gut is a lifespan limiting organ, it seems like, for flies. Maybe flies are essentially a tube, right? But they, it's not clear to me if that happens on a daily basis or if that's just something that happens near the end of life, or it happens with aging as well. You get sort of a buildup of oxidative stress in the gut. We saw that in our aging assays. Whether or not that's sort of cause or effect. Yeah, it's not clear.
So since aging has come up a couple of times, and it seems that aging is associated with a number of pathologies which are also linked to circadian rhythm disruption. So I wonder, like, what do you think is the connection between the circadian rhythm and aging, and what do you think are the kind of the big open questions about this?
Yeah.
Do you think there is a connection between circadian rhythm disruption and aging? I think this was something that came up in a number of talks where disrupting circadian rhythm might be associated with cancer or neurodegeneration, which are also thought to be aging-related diseases. But I wonder what kind of work that you've done on this topic. And I noticed that you had a paper which was surprising to me, which was that loss of certain clock genes actually extended lifespan, which I thought was, I mean, to me it was counterintuitive, but maybe it's not. I just
I think it was counterintuitive to everybody, including us. Yeah, that was hard to publish, honestly, because it does seem very counterintuitive. I mean, my only I guess what I would say is, I don't think of circadian rhythm, like circadian states, as good or bad, right? We think of them as just being different, right? So if the animal is oscillating between different states at all times of day, and shifting their physiology in multiple ways. What we just managed to happen to do with the period and timeless male flies was trap them in a state that had a particular effect on a specific physiology that is lifespan limiting in the fly. So I mentioned that the gut is a lifespan limiting organ. It's really important. Gut integrity is really important to the fly, and it's been shown that they'll lose integrity. This is David Walker's work. They'll lose their integrity, gut integrity, some number of days before they die. Many of them, not all, but a large fraction of them. So it seems as though that contributes to overall mortality in a pretty significant way. And what we found is that HER and TIM mutants are upregulated in mitochondrial uncoupling protein. It's a specific gene that in mammals is important for thermogenesis in the fat body. So it causes mitochondria to go through what's called futile respiration, right? And it makes a metabolic activity like very high. So these period and timeless mutants were eating a lot and really active and exuding a lot of carbon dioxide and being very metabolically hyperactive, right? And that had the sort of effect of decreasing or inhibiting proliferation of intestinal stem cells and decreasing oxidative stress in their guts. And that is what we think leads to their long lifespan. So in the fly, it very specifically has been shown that oxidative stress buildup in the gut leads to aging and mortality, and also leads to this expansion of intestinal stem cells and misdifferentiation, and changes the integrity of the gut, right? So you get leakage of gut contents into the hemolymph, essentially. And so you can get like a sort of an opportunistic infection. This is the hypothesis anyway for aging in the fly. And it just so happens that mitochondrial uncoupling protein will inhibit that process of aging of the gut somehow and extend the lifespan of the fly significantly.
No, I think, yeah. Does it matter when in the life of the fly you knock these genes out? Is there a way of doing it in adulthood versus development?
Yeah. So we did do it during adulthood, but we didn't actually play around with can we do it like late in life versus early in life? That's a really good question. We should try that. I mean, it's of interest, right? To like people who are aging already and can't go back in childhood. I mean, that's a really interesting question. We haven't really explored that as fully as we should.
Yeah. So as the last kind of question, I would be interested to hear kind of what are the next projects that you're interested in the lab, or what do you see as the next big open questions in the field around these topics?
Yeah, I mean, we like to sort of join things together. So, we're interested in sleep and oxidative stress and aging, of course. We're interested in circadian regulation of the immune system, and in particular, sort of chronic inflammation that occurs during aging. And we're interested in sort of dietary interventions that can change aging. So in particular, time-restricted feeding. So our most current work deals with time-restricted feeding, and hopefully it will shed some light on the underlying mechanisms that lead to health benefits due to time-restricted feeding.
Well, nice. Well, we'll look forward to hearing more about it when it comes out.
Thank you.
It was really nice to chat with you. I think we're at time, so we can kind of wrap it there. Unless, is there anything else that? Yeah. So I think we can wrap it up. Yeah. No, I think that was nice. It was really nice to talk to you.
yeah, thank you for having this conversation. It was really fun.